Hello, the Cancer Guy here. I'd like to talk to you today about two amazing new technologies, antibody drug conjugates and bispecific T cell engagers. I'm sure you've heard about both of these. They are radically changing how we treat cancers. But what's really important is how to determine which ones are going to be best for you. Now, there are many different ways of doing this, but we have a solution that we feel is one of the best for determining which antibody drug conjugates and T cell engagers are best and how to get into clinical trials. So, first, our disclaimer. Please read this. So, antibody drug conjugates and bispecific T cell engagers. As I mentioned, they have really been game changers in the cancer treatment industry. Both drugs are based on what we call drug targets. Drug targets are either highly overexpressed tumor cell surface receptors or tumor cell antigens. Basically, cell surface receptors are things that modulate and regulate what goes in and out of a cell. And they're usually used to identify tumor cells because tumor cells can have unique combinations of these, as well as antigens. Antigens are more like a street address of a house, and they identify a cell as a certain type of cell so the immune system can recognize it. Both are very important because when they're overactivated on the tumor cells compared to normal cells, they act as drug targets that can be used to target using antibody drug conjugates, bispecific T cell engagers, and all kinds of different monoclonal antibodies and small molecule targets. However, the testing to determine if a specific drug is beneficial based on overexpression of these drug targets is something that is very much a challenge right now in the clinic. Typically, what is used is a pathology-based approach called the H-score method. Typically, the H-score method starts with a biopsy sample, and a biopsy can have anywhere from 10, 20% tumor cells to 50, 60% tumor cells. That's a very important factor there because when you are counting cells, you're hoping that the cells you're staining are actually tumor cells. But obviously, having a diluted sample can be a real problem. I'm not going to get into laser capture microdissection, which is what we use for all of our tumor DNA and tumor RNA samples. There's some great videos on that. You can Google it, LCM. We have some videos on it too. Basically, it's just a process where a laser beam is used to isolate the tumor cells out of a biopsy sample, and that biopsy sample is then tested because it's going to be purified tumor cells. So the H-score is calculated as 1 times the percentage of weakly stained cells, 2 times the percentage of moderately, and 3 times the percentage of strongly stained. And it results in a range of 0 to 300. A low drug score is less than typically 100, and a high drug score is typically 100 and above. Now, there's a lot of variation on this. Different cell surface receptors and antigens have different cutoffs. So this is just a generalized approach of determining antibody drug conjugate and bispecific T cell engager targets. However, as I mentioned, there's a much better method, and that's referred to as tumor RNA expression testing. This approach counts the number of drug target molecules being produced by the cell. In other words, the RNA molecules being produced by the cell in the tumor and compares them to levels found in healthy normal tissues and comes up with a fold change. In other words, how much is that particular marker overexpressed compared to what it would be in normal tissues? While the H-score uses a 0 to 300 score to quantify the intensity and percentage of cell surface staining, fold changes measures the upregulation of the drug target messenger RNA compared to normal or control tissues. So we get a much more accurate assessment of drug targets across various tissues, or that can be predicted across various tissues. And this means that we can somehow or in some way get some sense of how the drug is going to work in various tissues based on their relative expression level. So the benefits of tumor RNA expression testing is that you can look at many different molecular markers at the same time from a single sample. With H-score, you have to redo the testing for each new molecular feature, and you quickly use up your tumor sample. So, most biopsies, you could probably only do maybe 2 to 4 or 5 different H-score samples. And considering the amount of different antibody drug conjugates and bispecific T cell engagers that are available right now and all the different targets, there's probably close to 40 different targets now. You want to be able to have a system that looks at as many different targets as possible. So our tumor RNA expression panel is actually looking at just under 21,000 different drug targets in a single sample. So your one little biopsy sample can give us a massive amount of information. And this is important because this can tell us which drugs are going to be potentially useful for you as they come out. So, when we look at all these targets, we can turn around and say, well, there's no target for this particular marker. And then a few months later, we'll say, oh, there is a target now, or there is a drug for this marker now. This drug can be used for you. So the problems with the H-score is it can misinterpret tumor cells, primarily due to factors related to what we call intratumoral heterogeneity, which is variation between tumor cells, technical staining variability, which is the actual application of the stains they use in the H-scoring method. And in the case of automated systems, limitations in distinguishing weak staining tumor cells. In other words, cells that are weak staining but still could act as a potential drug target. So intratumoral heterogeneity. Since tumors are not uniform, some areas may have high protein expression while others have low or no expression. If the H-score is calculated on a small biopsy sample that does not represent the entire tumor, then you're going to have different expression levels that could be missed throughout the tumor. Additionally, weak staining cells, automated analysis may fail to detect or improperly categorize tumor cells with weak staining, which would be a 1+, leading to inaccurate, usually high H-scores that are not representative of the true tumor samples. There's also the issue of staining intensity thresholds. The H-score depends on classifying cells into none, weak, moderate, or strong staining. The cutoff thresholds may not catch all cancer cells that express a protein, let's say at a very low level, potentially misclassifying them as negative when they really could have a response to a certain drug. There's also antibody limitations. Some tumors still retain protein expression and can be missed by immunohistochemistry or IHC-based testing, just because the antibodies are not a perfect fit for that receptor or antigen. You can also have issues with pre-analytical variables, things like tissue fixation, time and formalin, the preparation of the sample. Was your biopsy sample prepared in Canada? Now, we're supposed to follow the CLIA and the CAP protocols, but I have to say in Canada, rarely do I see the tissue fixation time accurately represented, especially on a weekend or something. Actually, I wouldn't say rarely. I just say we don't see it a lot. A lot of times it's not even reported. Sometimes it is reported, but it could be a real problem because then we don't know for sure if that sample has been prepared properly. And this can cause inconsistent staining, leading to inaccurate H-scores. Also, the detection of drug targets with H-score, you are potentially going to be on a drug trial that isn't right for you, or you could be refused access to a trial that could benefit you, as well as missing out important information regarding many other treatment options because your tumor sample has been used up testing for 1 or 2 clinical trials or 1 or 2 drugs. So this is a real problem. I urge you, if you're considering looking at antibody drug conjugates or bispecific T-cell engagers, or you want to enter a clinical trial, use tumor RNA expression testing. So here is our plug. You can get one-on-one educational consults. We've got a new 30-minute fast track that you could book yourself. Now, once again, these are educational-based consultations in order to educate you on what is available and to help you access that on an international level. We are not prescribing, we are not treating, we are not diagnosing. We are merely letting you know of all of the potential drug treatment options and therapies on an international level. So I highly recommend you get a consult with us. Or check out our YouTube video, and Michelle has a wonderful precision oncology education and advocacy program called Just the Facts. Have a great day.